I'm going to turn that off. Hello everyone, my name is Parveen. I am a senior lecturer in the now new cardiovascular department. And previous to that, I was in pharmacology over in ITM. And before that, I was a postdoc at the University of Toronto in Canada. So I'm going to tell you today about some of the work that we in my lab have been doing. And we have kind of focused on looking at cardioprotection, and we're looking at the problems and hopefully finding solutions to some of the problems that I'm going to talk to you about today. Okay. So today I'm going to give you a bit of a background, a bit of a clinical perspective on why this is a problem and why we need to be looking at it. We're going to talk about cardiotoxicity. I'm going to introduce briefly some cardio-oncology that is going on in the group, and look at some of the model systems that we're using to try and get our research to be as in vivo-like as possible, and introduce the NRF2 pathway, although probably you've heard about that from Vanya. I know Vanya does a lot of work in the NRF2 in this department. And we're trying to see if we can highlight how NRF2 could be used for cardioprotection. So coming from the Department of Pharmacology, you can imagine we were interested in a lot of drugs. And one of the things that is quite clear is that one of the major concerns and one of the major reasons for drug attrition in development and the removal of drugs that have been approved from the market is cardiotoxicity, and the second being hepatotoxicity. So there's quite a big reason in the field of drug safety to look at both cardiovascular and liver toxicity together, which is what we were kind of doing in the other department. And so I have a PhD student who works on the hepatotoxicity side of things, and we now have a couple that work on the cardiovascular as well. So looking at this, we can see that going from non-clinical all the way over to post-approval, that the cardiovascular system can cause problems such that drugs are, stopped either in the non-clinical phase or going into phase 1 all the way up to phase 3, and then pulled from the market. And these can be very problematic, and we need to be able to understand why the cardiovascular system does take such a hit from drugs and what we can do to try and prevent this toxicity. And the drugs that we're talking about, some of them are quite specialized and some of them are actually quite commonly available. Drugs that can cause cardiotoxicity are kind of divided into two different areas: those that are on target, so those that are given for conditions that are related to cardiovascular complications, such as antiarrhythmics, which are prescribed if you have an irregular heartbeat. And these can lead to acute or even chronic toxicities that can make that existing arrhythmia worse, or actually even sometimes lead to other arrhythmias arriving because of the toxicity of that particular medication. One of the problems that we tend to look at is off-target toxicity. So we've got here medications that are given for cancer. So these are things like anthracyclines, which are chemotherapeutic agents. And I'll come back to these for the majority of the talk, but also things like alkylating agents, which are again given for cancer and autoimmune disorders. And these can lead to either very acute problems within, say, 2 weeks, or these can build up over many years and produce cardiovascular damage that isn't actually apparent until about 15 years after the treatment has been withdrawn. So it's not often clear that that person has got a cardiovascular complication until many, many years after that initial hit of the chemotherapeutic agents. And one of the kind of more concerning medications that are, or class of medications that are causing problems that aren't really prescribed for anything, you know, as life-threatening as cancer or irregular heartbeat, at the non-steroidal anti-inflammatory class, which are prescribed for pain management. But as the population ages, we're seeing the need for you know, managing chronic pain such as arthritis. And so people have prescribed NSAIDs for many years to manage the pain from such chronic conditions. As we live longer, this is even longer than it used to be, and this is leading to the detection of more cardiovascular complications like myocardial infarction and stroke. And so these drugs do remain on the market. These drugs are not going to be removed because they are very effective at alleviating the primary concern. You know, they're very good at what they do, and the side effects are going to have to be managed. And we need to find a way to minimize that level of cardiac damage and have early detection, because that's the way we're going to get around it. We're never going to be able to remove these medications because they are too good at what they do. So what is cardiotoxicity before I launch into a talk about cardiotoxicity? It's defined as a severe and potentially fatal adverse reaction to certain drugs. It can be chronic, can be acute. It can be caused because of on and off target conditions that we talked about in the last slide. It can cause structural changes, and it can affect the functionality of the heart, and it ultimately reduces its efficiency to pump blood. And it can cause changes at a cellular level that then lead to its functional dysfunction, causing left ventricle ejection fraction compromisation. And this parameter tends to be what's monitored when patients are first diagnosed with cardiovascular complications due to drugs. It can then lead to things like arrhythmia and all the way over to heart failure. So just to look at that parameter that does get measured, the left ventricle ejection fraction is the measurement of the amount of blood pumped out of the ventricle over the total amount of blood. And so this gives us a value that can be measured through echocardiography and give us an idea of how well the heart is actually functioning. So normal healthy people have an ejection fraction of around 55 to about 70%. But cardiotoxicity is then defined as the decrease in left ventricle ejection fraction of more than 10% to a final value of less than or 50%. Anything below that, so about 40%, is indicative of quite severe heart failure or cardiomyopathy. So, as I mentioned, because we're starting to improve patient health with the cancer drugs, and they really are turning into almost a victim of their own success. Patients are living longer, and we're starting to see the development of a major side effect in that a lot of the cancer drugs that are out there are showing long-term effects of cardiotoxicity. And these include things like chemotherapeutic drugs such as doxorubicin that we will come back to, but also targeted therapies such as Herceptin, and even the newer, more recent checkpoint inhibitors. And we're seeing with those, it's not very common to see things like myocarditis, but in those patients that do get it with the checkpoint inhibitors, it can be quite fatal. And it's becoming a problem that needs to be addressed clinically. And so much so that after secondary malignancies, the leading cause of death in cancer survivors is cardiovascular disease, which actually affects about 1 in 10 patients. And so it is a real concern, and we do need to address this. And as I've said before, we need better ways to predict it and prevent it, as it's unlikely that these very efficient anti-cancer medications are going to be withdrawn because they are such good at dealing with the cancer. We're not going to, the primary concern for a physician who's dealing with a patient's cancer is dealing with the life-threatening condition. It's not thinking in many years that they will have to deal with a cardiovascular complication. But in the same respect, you know, we can't have patients who have battled and fought with cancer then having to then fight a second life-threatening condition. So, you know, this field of cardio-oncology, to some degree, unfortunately, is quite rapidly developing, and there's a lot of cardio-oncology units that are being developed, and we work closely with the cardio-oncology, the newly formed cardio-oncology department at the Liverpool Heart and Chest Clinic. So the drug that I'm going to talk to you about in the rest of my talk are the classes, the anthracyclines. These are chemotherapeutic agents, and the most commonly used is doxorubicin. And this is a first in-line therapy for multiple cancers, including things like leukemia, breast cancer, lung, ovarian. And it's associated with a well-known dose-dependent cardiotoxicity. And the problems arise because of cumulative doses of about greater than 500 milligrams per meter squared. And it can lead at that dose to affect, you know, between 20 and 25% of patients having cardiovascular complications. And this, you know, is a concern in all the patients, but if you can imagine, if you're a patient who is in early childhood that may receive quite a high dose of doxorubicin, and then you do have a secondary malignancy, then that accumulative dose, the physicians will try their best to keep it below 500 milligrams. And that may then mean that you have less of this treatment, because there is a real concern that, you know, somebody so young will have lifelong problems if that dose is exceeded. And the statistics are actually quite shocking in that they say that the numbers come in at that 57% of children that have been exposed to anthracycline 6 years after they've survived their cancer, 57% of them have some form of subclinical change in their left ventricle structure and function, and this is detectable by the echocardiography. And so, especially for very young patients where this therapy is very effective, we can't have such devastating cardiac problems quite early in their adulthood, if it's only a few years after the end of their chemotherapy. So what can we do currently? So currently we're using doxorubicin. As I've mentioned, it can be, in some patients, it causes no complications, but in a lot of patients, it can cause complications. And at the moment, the only FDA-approved drug for cardioprotection is a drug called dexrazoxane. But dexrazoxane itself is associated with secondary malignancies. And so its prescription is actually limited. Its administration used to be, up until last year, only to stage 4 breast cancer patients. And more recently, they've included in that subset children and adolescents who receive a high cumulative dose. And so it is it's a drug given to a limited population of people, and it's not rescuing them completely. But that does leave then a wide range of patients that have no cardioprotective drug to their disposal, because dexrazoxane is not actually given to a wide range of people. So that's where we were kind of trying to come in. What can we do? Where can we take our research to better provide cardioprotective agents and cardioprotective mechanisms to protect people who may not be able to receive the dexrazoxane? And one of the first things that we have to do with cardiac research is actually find a good cardiac model system to look at anything that's going to give us a translational result. And the problems with cardiac research arise from the fact that the heart is it's a non-regenerative organ. It's got very limited potential to heal, to a point where you're very unlikely to be able to get any kind of healthy tissue from a patient. Nobody's going to give you a cardiac biopsy from a tissue that doesn't heal itself. Nobody in their right mind anyway. And it's not like what we do with the liver work, where we have patients coming in who might have a liver complication. They'll give you a piece where it's damaged, and then they're quite happy to give you a piece where it's not damaged, because you know that you can take out 2/3 of the liver and it will grow back. The heart does not have that potential, and so gaining primary tissue is actually quite difficult. We heard last week with Richard's talk that animal models are an alternative, and you can use animal models quite efficiently to look at whole in vivo like experiments. But we've, in the beginning for now, started to look at cell models. And so for cell models, there are cell lines that are available. These are usually immortalized lines that will divide. And so the advantage of these lines is that they do divide, that we've got lots of material to work with, but the disadvantage is they don't tend to beat, they don't tend to have the exact morphology of a primary cardiomyocyte. To kind of get over that, there is now the use of either embryonic, it used to be embryonic stem cells, the move now is to induced pluripotent stem cells, and those cells will provide you that phenotype where these cells will beat. And we can make the iPSCs, and this is some that we made whilst I was a postdoc, and they will sit in a dish and they will beat, and these are therefore better in a way than the cell lines, even though the cell lines are useful in the beginning when you're doing the initial phases of research, because growing iPSCs is still quite expensive, unfortunately. The problem with the iPSCs, however, comes from their maturity. So this is a sheet of iPSC-derived cardiomyocytes that we can grow in the lab, and it will grow, but no matter for how long you grow it, it will remain as this kind of sheet that retains a very neonatal-like phenotype. This, on the other hand, is an adult cardiomyocyte, and you can clearly see that these two are worlds apart. We cannot get this, at the moment, neonatal phenotype to look adult-like. And there's a lot of effort actually going into trying to develop this maturity aspect of it. I work with one of my PhD students is in Pfizer in America, and one of the ways he is looking at maturing the iPSCs is to electrically pace them. And so the idea being that if you induce pacing in these cells, that will increase their maturity. And so that's one area and one method that people are trying. A second, working with colleagues still in Toronto, is mechanical stretching. And we have colleagues that grow these cells on biowires that then stretch out the cells. But unfortunately, that is actually quite a technical process. And that team work very closely with bioengineers that create these scaffolds that then can be stretched. And therefore, you need quite a lot of additional techniques to be available to be able to try that type of cellular maturation. So the way we've done it, when I started here in Liverpool, was to use 3D spheroid systems. So when I arrived here, Dr. Mike Cross, who is in the department, had developed a 3D model system that we're now using and developed a little bit further. And so what we had done is taken cardiomyocytes, the iPSC-derived cardiomyocytes, which are from the heart, and also added to that cardiac fibroblasts, which make up the matrix of the heart, and cardiac endothelial cells that then that then line the blood vessels. And we're then able to mix these together to make a mixed spheroid, but also a single cardiomyocyte type spheroid. We take these cells, we grow them in ultra low adhesion plates. We can then grow them individually. We then put them together at specified ratios. Again, this was the work that Mike had done when I arrived. And we allow them to form, and we get a single spheroid in a 96 wall plate. Our little spheroid, this is what they look like. We can add them, and they beat after about 2 weeks. So just comparing the mixed spheroid to the cardiomyocyte only one, what we found is that the cardiomyocyte only one didn't really beat to the same degree as that mixed spheroid, kind of suggesting that you needed that cellular crosstalk. And so this is an image here of our mixed spheroid that beats after 2 weeks. Excuse me, the cardiomyocyte only one doesn't look quite as good. And if we actually visualize them, what you see with the cardiomyocyte only spheroid is that it tends to form a very loose structure, and it's not very well maintained, and they're very quite difficult to manipulate and to move. So we kind of then, you know, if we look at the mixed spheroid, however, you can see they form these nice balls of cells. They hold very well. You can move them around. In order to do the immunofluorescence, for instance, you need to wash them and add antibodies and then rewash them. And they're quite, you're able to do that. They're quite robust. The other thing with the mixed spheroid is that you can see that they do start to then take on the striations that we saw with that adult cardiomyocyte that I had. And you start to see the formation of striations within the spheroid. So they are taking on a more mature phenotype, which is exactly what we wanted to do. Over time, you can see that this is at 1 week and these are at 5 weeks. You can see that all the cell types are retained as we go through over periods of time. We've used cell-specific markers of, you know, PCAM for endothelial cells, collagen for fibroblasts, and the Hurst is just for nuclei. And you can see that they're retained over time, all the cell types through this spheroid. So, you know, this is where we started off with the iPSCs or the embryonic stem cells. This is kind of where we want to end up. We're not there by any means, unfortunately, but we are somewhere in the middle. We have got this 3D model that has got the structural phenotype that's more similar to the adult. This is an electron microscopy that we've done of this spheroid, and you can start to see characteristics of a striated cardiac tissue, forming. And, you know, improvements still need to be made where we're not generating cells that look like this, but we're further away than we were. So it is improving. We've looked at this as well through some mass spectrometry. So here in blue so this is an experiment where we've taken the spheroids at week 1 and then at week 5. And in blue are proteins that don't change, and in red, sorry, I've lost my mouse. Oh, in red, we've got proteins that are significantly different. So significantly upregulated here, significantly downregulated here. And if we kind of look at this in depth, then in here, we've got proteins that are involved in cardiac contraction, things like phospholamban, the ryanodin receptor, the sarcoplasmic reticulum. And also, interestingly, a protein that we found in Canada to be involved in cellular maturation. So this protein here, TMEM65, was something we did a lot of work on because it was one that we found in a proteomic screen to be elevated in mature tissue compared to the neonatal cardiomyocytes. And we find that it's actually involved in contraction. And so that was really good for us to be able to see an enrichment in that as these spheroids age, to give us more confidence that we're actually seeing cellular maturation. Looking at the proteins, just looking loosely at those that were differentially altered in here, then we see an upregulation here in red in those proteins that are those proteins, we see an upregulation in proteins that are involved in heart development, in muscle contraction, in ion transport, so proteins that we expect to go up, and a downregulation in proteins that are in, cell cycle, cell morphogenesis, because cardiomyocytes in the heart are terminally differentiated. And so we expect these again to come down. So we are improving as we go along, and we hope someday to get to this, but we're at least getting closer. So having got a model system that was looking more in vivo-like, we then started to look at the pathways involved in doxorubicin-induced cell death. So doxorubicin can enter the cells either passively or through certain transporters and channels, is known to cause cell death by multiple mechanisms. It can cause lipid peroxidation, it can cause mitochondrial dysfunction through the generation of ROS, which then the reactive oxygen species then cause mitochondrial dysfunction. It can bind topoisomerase 2, therefore cause breaks in DNA transcription, and all of this will ultimately lead to cell death. Dexrazoxane is an iron chelator. It prevents, it sequesters iron, which then prevents this formation of this semiquinone metabolite that then leads to production of more hydrogen peroxide. And that's how cardioprotection up until now from anthracycline-induced toxicity was actually being handled. What we've wanted to do is use now this NRF2 stimulation, use this pathway to directly prevent reactive oxygen species. And we've stimulated NRF2 through the use of CDDOME. So as I probably you've heard from a lot of this from Vanya already, NRF2 is termed master regulator of the antioxidant response. So I'm not going to go into too much detail on NRF2, but essentially it is a transcription factor that is sequestered to a protein called KEAP1 in the cytosol, and in normal non-stress conditions it gets ubiquitinated and therefore degraded, and it kind of sits in this cycle where it's bound, not doing very much, and then is degraded. Under situations of stress and under upregulation with CDDO, NRF2 is then sequestered into the nucleus, where it is bound to the ARE element, where it increases the antioxidant response. So CDDO is a synthetic peptide, actually, that when I was putting this talk together, I realized it has actually just recently been granted FDA approval for chronic kidney disease. And so that's kind of good for us. It means its safety profile has been looked at, and we would then be trying to repurpose something that's already got FDA approval. So we have this ball of cells, and the first experiments that we had to do was to really make sure that the response that we were seeing was from a homogeneous population. It's not, we've got this ball of cells. We needed to make sure the cells on the inside were seeing the drugs as well as the cells on the outside that were in contact. And so we've taken advantage of the fact that doxorubicin actually autofluoresces in the red channel, and we've used the light sheet microscope that's available in the imaging facility with VLA, and that microscope has the potential to then scan the entire depth of that spheroid. And so going from one end all the way through the middle to the other end, looking to see if this red fluorescence, which is indicative of doxorubicin, penetrated the entire spheroid. And you can see here clearly that it does in all dimensions. And here we've just superimposed that with our phase contrast image. So that was good. So we've got a system that will give us a homogeneous reporter on the drugs. We carried out a dose response curve, and we've got, as we would have hoped, emulating what happens in patients, a dose response curve that shows a decrease in cell viability upon increase in dose of doxorubicin. So as we increase the dose, the viability of these cells decreases. We added dexrazoxane to see, do we get that same patient response? And again, we see a shift in this curve, which suggests to us that we are getting to some degree of protection with the dexrazoxane. Now, this was actually quite big because people have tried to do doxorubicin studies before in iPSC-derived stem cells, but they've done it in 2 dimensions using flat plates. And up until now, we've not really seen any other report that's reported that dexrazoxane was being protective. People have been quite honest, actually, and said that they've not been able to rescue this damage using this current FDA-approved drug. So for us to be able to see it is very promising. It's very much in vivo-like, and kind of suggests to us that it is looking more and more like a translatable in vivo system. Not only do we see the same functional changes, but we also do see structural changes. And I showed you before that the spheroids have this striated phenotype that then gets lost as the dose of doxorubicin increases. We've added then the CDDO, and we've seen an increase in proteins that these are just some downstream targets of CDDO. We just wanted to make sure that we were seeing the right pathway being stimulated. But as I said, CDDO is not something novel. It's not a novel NRF2 protectant. It's just something not been used for cardiovascular and was in clinical trials when we first started. And we can see here that CDDO increases it. And quite surprisingly, we weren't really expecting this because dexrazoxane itself doesn't elevate the levels of these downstream NRF2 genes. But when we add them together, it causes a synergistic effect, and we don't really know why. But we do see this synergy between these 2 chemicals. And so we dosed our cells, we incubated it with CDDO, and what we see here is that this is the viability of the cells, and we've measured this using ATP viability with CDDO protection. This is with dexrazoxane protection, and this is with them both added together. So the 2 things to kind of take away from this is that CDDO protects the 3D system to the same degree as dexrazoxane. But when we add them both together, it does give us this synergistic response. And again, we're not really sure why this is happening, but we do see it over and over again. So the takeaway message was that CDDO was protecting to the same degree. We then looked at reactive oxygen species. And again, we've got that same degree of protection offered by CDDO elevating the NRF2 pathway as we do with dexrazoxane. We looked at this through the structure of the structure of the cardiomyocytes. And again, this is the untreated, this is DOX treated, and this is actually both of them together, the DOX and the DEX and the CDDO. And we see here, you get this retention of the striated phenotype. So could CDDO be offered to those patients for whom dexrazoxane was deemed too risky? And this is something now that we're trying to develop further, and we're trying to really look into, because it is starting to look quite promising. One of the things we were very, very aware of is that CDDO is just a general inducer of NRF2. It's not targeted to the heart. And the one thing that we should we should be all aware of is that we were not protecting the cancer cells at the levels of NRF2 stimulation that we were inducing. And so we've done this with breast cancer cells, these AU565 breast cancer cells, and we've shown that at the dose of CDDO and the duration of CDDO that we use is not rescuing the viability of the breast cancer cells to any degree from the doxorubicin damage that we want it to do. So there's no protection offered to the breast cancer cells at 2 different doses of doxorubicin in this graph, which is good. We don't want it to be protecting the cancer. We do want it to be protecting the cardiomyocytes. This is only obviously one cancer cell line. We are moving this forward to look at a variety of cancer cell lines just to make sure that any of the dosing that we're doing isn't affecting it. And also there are cancers that have elevated levels of NRF2. So what happens to those? And that's something we will do as we move along with this research. So then at this point, we started talking to Richard. He had moved from Leicester to Liverpool, and he kind of stressed on us the importance of glucose. And we heard last week from him that having elevated levels of circulating glucose can be detrimental to any kind of cardioprotective efforts that you are trying to use in the cell. High levels of glucose have been shown to, and he's shown it, others have shown it, to decrease the effect of cardioprotective agents. So we thought, you know, we better look at this and check this out. And we then had a look at our protective effect of CDDO in both high and low glucose. So in these graphs, high glucose is 20 millimolar, low glucose is physiological, around 5 millimolar. And although this says high doxorubicin, it's not. It was high compared to another dose that we were using for another part of the study. But the dose of doxorubicin used here is actually the same as the Cmax value. So say the same as the value that you would see in patient plasma. So interestingly, when we look at this, we're looking at ATP, again, using ATP as viability. With CDDO, we see a restoration, we see a rescue of this ATP phenotype only really in the low glucose. And here we've looked at an AC16, which is cardiomyocyte cell line, and these are human cardiac fibroblasts. And in both of these cells, CDDO increases ATP production only at low glucose, which, you know, is hugely interesting. We need to figure out this into more depth. Is it the case that the CDDO is protecting the mitochondria because we're reducing the reactive oxygen species level? Or is it a case that we're making mitochondria more efficient? They're using oxidative phosphorylation, which is a more efficient way to turn over ATP. And this is something we are following with one of our colleagues, Amy, who has Seahorse Facilities, which is a machine to look at mitochondrial functionality. And so we're waiting for those results quite excitedly. We did look at LDH production as well as a measure of cytotoxicity. And the LDH seems to be less affected by the glucose concentration. At both glucose concentrations, we see a decrease in LDH released with CDDO. So you see it here at the high glucose, but you also see it here at the low glucose. In both the cardiomyocytes and in the fibroblasts. So the CDDO is reducing the LDH levels at both concentrations, but only able to increase this ATP production at low glucose. And so we do need to be aware then of what glucose concentrations these patients are coming in with when they are receiving their therapy, because we're not really talking here about patients who have diabetes, because in those patients, the glucose is actually quite well maintained, quite well managed. These tend to be elevated levels of glucose that are caused by stress. You know, you've got patients going in who are receiving chemotherapy for cancer, they're going to be stressed, their glucose levels will go up. And so, you know, we do need to be aware that perhaps glucose does need to be measured and monitored before they receive any of this treatment. And one of the other mechanisms that we've been looking into, and again, in collaboration with Vanya, in collaboration with Professor Anne McCardle, who's also in the department, looking at cellular aging. We know that doxorubicin can cause damage in patients up to about 15, even 18 years post-treatment. So one of the ideas that we had was, is it causing premature aging? And so after speaking with Anne, she said, you know, the first thing to do was to look at the cells, see what's going on, and try using beta-galactosidase, which is a marker for senescence, cellular aging. So that's what we did. So we can see here images of the high and the low glucose, and quite startlingly, you can see here that you've got more cells in the low glucose. This is the same concentration of doxorubicin, but at this low glucose, there are just more cells. Interestingly, though, when we looked at the percentage of senescence, we do see, oh, I don't know what's happened there. We do see higher degrees of senescence in the low glucose. When we add CDDO, again, the cells are healthier, and what we see is a decrease in this aging process, so a decrease in the percentage of cells that are senescent, only in the low glucose with the addition of CDDO. We have tried this at 2 different concentrations of doxorubicin just to make sure this wasn't just a one-off. So this, again, the high is the Cmax value, whereas this is a subclinical dose. And again, what we see is that there is an increase in senescence at low glucose, but there are more cells. And this is, again, decreased with the presence of CDDO. So perhaps, and we've not looked into this yet, perhaps there is a protective mechanism, cells go into senescence so that they do not go into apoptosis, and this is a protective mechanism, but we do see that this is reduced with CDDO. And we haven't really kind of pulled CDDO and NRF2 out of thin air. We have been looking at NRF2 in the department for quite some time now, and looking at it through aging, again, is something that we've done in collaboration with Anne and with Vanya. And we've been able to get these mice that are aged to look at what happens with NRF2 and look at what happens when we knock out NRF2. So these are mice that Vanya had. She's been working on this NRF2 knockout model. And if we look at these mice as they age, so a 2-month mouse is a very young mouse. This is an equivalency kind of chart. This is the stage of the mouse. This is the stage of the humans. So a 2-month mouse is quite young. A 6-month mouse is about here, which is the equivalent of a 30-year-old human. An 8-month, you're kind of in the middle here. And a 26-month-old mouse is actually is an older person. And so as we age, what we can see is we've got an increase in markers of senescence. So our cells are aging, obviously. But in our knockout mouse, when we look at the 6-month wild type compared to the 6-month knockout, we see this increase in aging related genes and senescence markers, both in the atria and in the ventricle. So NRF2 is there in aging. You can see then here at 26 months old, we've got a huge elevation of these proteins. And so this 6-month knockout is somewhere in between here. We've not really done enough variations to figure out how much this has aged, but it has aged compared to its 6-month counterpart, wild type. So NRF2 is involved in aging. Knocking out NRF2 accelerates aging. So then hopefully what that means is then if we increase the amount of NRF2, we decrease aging. And so when we're looking at this diagram of cellular aging here, what we think is going on is that we've actually just reduced the amount of oxidative stress, which has decreased the production of proteins that are essential in aging. We're not suggesting that CDDO is a senolytic. We're suggesting that we're stopping this higher up in this flow. And this has also been seen in humans. And so this is just some geodata that we've pulled out of the NCBI. And we can see here that in human cardiovascular diseases associated with aging, so dilated cardiomyopathy, ischemic cardiomyopathy, we've got a decrease in NRF2. And I've got another student who's looking into some of the downstream proteins that NRF2 regulates to see exactly which proteins are downregulated in these mice, in the mice and in humans from this geodata at the moment. So if we bring this together, chemotherapy-induced cardiotoxicity is a major concern, both acutely and chronically. Mike has been working on this 3D spheroid system. We've matured that a little bit further, and it's easily reproducible, doesn't require any extensive technologies. It's multicellular, and it can detect both structural and functional changes upon drug incubation. And it emulates drug injury with doxorubicin, but also protection with dexrazoxane, which hadn't been seen before in 2D. And so NRF2 stimulation attenuates doxorubicin-induced cardiotoxicity in cardiac cells, but we do need to be aware of, we do need to be aware that glucose levels may affect that level of protection, and that some of the mechanisms that go with this long-term damage might be due to the promotion of early premature aging. So taking this further and on to the next steps, could CDDOME be a less risky option than dexrazoxane? And how will this fare in long term? All the results we've done so far have been looking at an acute model. But the advantage of the 3D model is that the spheroid system is actually viable for 10 weeks. So this is going to allow us to do a more chronic time-based study with repeat dosing as seen in patients. So patients are given doxorubicin in 3-week intervals. And because of the time that we've got with the spheroids, we can now start to emulate this. We needed to be sure that what we see is, somewhat protective. So we've done the acute study, but now we're going to launch into looking into a more chronic study and look at more of the mechanisms over time to make sure that what we're seeing does actually help in a chronic time as well as an acute. And finally, I thought this talk was going to be too short actually, and now I'm a little bit worried about time. The last couple of slides I'm going to spend on another way that we're looking at doxorubicin-induced toxicity. So as I kind of mentioned in the beginning, we are working in collaboration with the cardio-oncology team at the Liverpool Heart and Chest. And one of the grants that we've got recently has been to look at more of a personalized medicine route. And we're all aware that we can take samples from patients. These can be blood or skin. We can add reprogramming factors, make our own iPSCs, and create cardiac myocytes. And so, this is a well-known now mechanism, and we are trying to bring this to Liverpool because at the moment, I don't think there's really that many groups that are doing their own iPSC programming. And so we're starting to do this in my lab. And the idea is that we will then take patients who have been on cancer medication with symptoms, but also those that have not had any symptoms, take we're actually going to look at blood cells, reprogram these, and then have cells that are from patients with damage and those without damage, and really kind of look at them in depth through multiple techniques to try and understand from that side of things what's happening in these patients. And so we actually started this, unfortunately, in January, and then the lockdown happened and we haven't got as far as we'd hoped, but we are starting to now pick this up again, and hopefully, fingers crossed, everything will be fine and we can actually get on with this. And so the overall concept of what's going on in my group is to gain insight into drug-induced cardiotoxicity, to provide potential drug targets, early detection biomarkers, to kind of look at CDDO and if it's effective and can be given to those patients that dexrazoxane might not be able, that might be too risky for them to have, and potentially provide another cardioprotection drug to a growing clinical problem that continues to be a concern, because as the population ages, unfortunately more and more people will have to go on these quite toxic medications, and so we do really need a way to protect the heart of these patients. And thank you to everybody on here, Liverpool, our collaborators, both, the Liverpool Centre for Cardiovascular Science and at Liverpool Heart and Chest, and our funding bodies. Thank you very much.
Oh, Parveen, thank you very much. That was really interesting, and that actually has generated a load of questions in the chat box if you want to open it. But I'm going to start since I wrote the first thing in there. So we do actually have some ways of stretching cells if you're interested in doing that. We have some of these cell scale systems where you can use your own matrix. It's not like the FlexiCell system where you're stuck with using a silicone rubber sheet, but you can use your own systems. And potentially, if you're interested, we might be able to, we have some really nice gels which we can possibly embed your spheres in and then stretch them afterwards, if that was of interest.
Oh, that sounds really interesting, yeah.
Okay, so we'll talk about that offline then, but it's something that we're thinking about with some other projects. So we can also make our gels with different mechanical properties. So
yeah, that sounds really interesting. Yeah, I'll follow that up with you afterwards, please.
Okay, very good. Paul asks, can you paste the cardiac spheroids?
You can. They're very small. We haven't tried it, but I think Mike has been looking into oh Mike, what did you call it? There's a system where you can paste them, that you can put them in. I think you can. Yes is the answer. We haven't done it.
Okay.
Yes, we have, Parveen.
Oh, we have?
Yes.
Who's that, Rich?
Yeah, we did it in our lab, if you remember, just before lockdown. We can get them to work.
No, so you were doing the electrophysiology. Did you actually paste them?
We did, yes.
Oh, I didn't know that.
Very good. Richard, do you want to ask your question while you're online?
What was my question?
Tox
oh yeah,
causes structural changes.
Does it cause, you said it's caused structural changes, does it collapse the T-tubule network in these cells?
I don't know. It can damage the sarcomeres, because then that translates to the reduction in the left ventricle ejection fraction, because you haven't got that force. Whether it's specifically T-tubules, I don't know. Because
it looks like the T-tubules are pretty much gone from your images, and that's something that happens in heart failure. You can lose those T-tubules, so that you could be getting drug-induced heart failure just from collapsing the T-tubules.
Could well be, yes.
Yeah, just interested.
On the opposite, does the CDO, if you put that in with your spheroids earlier on, would that help them to mature? Because they seem to get the better striation with it.
They do. They are. The CDDO will be protective, but I think a baseline, there's not that much stress on the cells, so they don't actually benefit from having the CDDO. It's not detrimental, but there's no added bonus of it, because they're quite happy as spheroids. They beat away. They're quite fine as long as we feed them and don't mistreat them too much. They're usually quite fine. But no, we don't see added maturation because of them.
Okay. Vanya, you have a couple of questions. Do you want to come online?
Oh, I'm glad you're here, Vanya.
Hello,
Vanya?
Vanya?
Can you hear me? Sorry.
Oh,
Yes,
yeah.
we can.
Good.
Okay. Yeah, very nice talk. I just wanted to ask, because we actually see that CDGO can increase an antioxidant response differently depending on time of day. And just wondered whether there is any rationale to maybe check the optimal time of day for this combined treatment. Yeah, that was my first question.
Yeah. How is it improved with time of day? If you don't mind me asking you a question.
Okay, so so if we, for example, administer CDDO, like say morning versus evening, and then check for antioxidant gene induction at those different times. So say, for example, 4 hours after treatment or 24 hours after treatment at 2 different times a day. We find that, for example, CDGO can be like something like up to 3 or 4 times more effective.
Oh.
Yeah.
We usually leave our CDDO in for 20. We leave it in for 24 hours before we treat the cells with doxorubicin.
Okay.
So
okay.
We've not looked at time of day differences, if I'm honest.
Yeah, I mean, to do that, you'd have to synchronize those, for example, cell phosphorylates beforehand.
Yeah, how would you tell a cell system what time of day it was?
So there is a couple of different drugs that people use to synchronize the cells in vitro, and then depending on a certain molecular clock markers, you are able to know how that relates to in vivo, if you see what I mean. I've also seen that some people often talk about this time of day treatments for cancer treatments, because they say there are times when you can reduce toxicity and you know, or for example improve efficacy when it comes to like killing the cancer cell. So it seems like an interesting area for this type of work. And my second question was, is it known much about the molecular pathway which leads to this cardiotoxicity
Of doxorubicin?
Yeah.
The molecular pathway, I mean, there's a lot of work that goes into looking at antioxidants, but general antioxidants don't seem to be very effective, which is why an RF2, which is like a master regulator, is probably more effective, or at least, yeah. So it is well known that it damages mitochondria, it can bind to DNA, but I don't think we know the full picture, which is why we can't fully protect it.
Is it at all linked to ion channels?
We haven't looked into it yet. That's the kind of next phase that we're trying to do with Rich.
Okay.
That you know, we put these. It was barely, I think it was a week before the lockdown, we had started to generate a large batch of the spheroids to look at a time course, and then lockdown happened, and I think Rich had a go with a few of them, and we were just starting to see calcium changes in the spheroids, but we don't know to that depth yet, Vanya.
Okay, and my last point was that we also have knockout cardiac tissues from 8 to 20 months, but not at 26 months as far as you went with wild type tissues. So that might be useful.
That would be very useful. Yes, please.
Yeah, and just the point about the mitochondria, we find higher mitochondrial activity in cells with low glucose.
Great. I see Sammy's on here as well. Sammy is the expert in the seahorse world, and I know, are you there, Sammy? No.
Yeah, come online if you're here still. I think maybe she may have had to leave.
Yeah. So we are going to be talking to Sammy and her former boss, who actually share an office with Amy, that we're going to be looking at the effects of mitochondria, and and if we see higher mitochondria activity at low glucose, then that really is a reason to try and promote low glucose before doxorubicin is given. You want your mitochondria to be as healthy as possible, so why wouldn't you do it at low glucose?
Yeah, it makes a lot of sense.
Yeah.
Thank you.
Thank you.
I think, is there any other questions? If not, I think we probably should call it a day, because we're actually just over 2 o'clock, and I suspect people need to get to other meetings.
Okay.
Any more questions? Anybody? Well, thank you very much, Parveen. It's very interesting to see how this is developing, but I think there are some areas where we're also doing some things with ex vivo models with different cell types and things like that, where there might well be some other areas where there's crossover, and things that we've managed to make things work that might help you, and things that you're doing might well help us as well.
That's
I think we should definitely keep conversations going.
Yes, definitely.
Great, thank you very much, everybody.
Thank you very much.
And hopefully see you next. I can't remember who's speaking. I think Helen Wright's speaking next week. Very good. See you all soon.
Bye.
Thanks, Parveen.